Molecular markers. So when scientists want to investigate the genetic diversity of a population, they have several different options because there are different techniques for studying DNA and there are different types of DNA. Remember, some of the DNA is in the nucleus and some in the mitochondria. We're going to talk about a few different types of molecular markers and what they can be used for. The first one we're going to talk about is mitochondrial DNA. We've already talked about how there is DNA both in the nucleus and the mitochondria. The DNA in the mitochondria helps power the cell and is therefore highly conserved, meaning that it doesn't change much over time, because if it changed, cell function would cease and that would mean problems for the individual. Because mitochondrial DNA doesn't change much over time, this makes it very useful for certain things like identifying species. Mitochondria is made up of several different regions, and these regions are generally found in all living things. These regions help code for important cell functions and therefore generally stay the same. But over time, a few mutations here and there occur, and the code between species is therefore slightly different. So this type of DNA is the DNA that is used to barcode individuals and identify species. When I talk about barcoding, what I mean is that we can identify different species using small segments of mitochondrial DNA. We take a short segment of the DNA and sequence it, and then compare the sequences, and we can ID species. So for example, I'm currently working on a project with two different species of rail. Here we have a picture of them. The king rail is on the left, it's a little bit more red, and the clapper rail is on the right. Both of these species look the same, and they live in similar marsh habitats, but they are found along a salinity gradient, with the king rail found in more freshwater systems and the clapper rail found in more saline or saltwater environments. Let's take a look at a specific primer called ND2 of their mitochondrial DNA. Okay, so here's the actual DNA code. The first few of these are clapper rails, and the last few are king rails. It can be hard to see the differences with all these letters, so we can tell the program to put a dot where everything is the same and show letters where there are differences. You can see here that there are a few letters that are different. For the clappers, they have an A, and then the kings have two differences. They have a T and then an A. So if you test this across many different individuals, and then the clapper rails always have that A, and the king rails always have that T and that A, then this variation is diagnostic of species, and we can use this to identify species. So this is really useful for species identification. But as you can see, every individual within the species has the same code. So this type of marker is not useful for identifying genetic diversity in a population or trying to identify individuals for things like population estimation. Also, we only get mitochondrial DNA from our mother, so it only tells us about half of the lineage. But it's commonly used in phylogenetics, where the differences in the mitochondrial sequence variation is used to map an evolutionary tree depicting ancestry. The next marker I'm going to talk about is a microsatellite. This is known as a simple sequence repeat. It's usually 2 to 5 base pairs long. So for example, here's our DNA code, and you can see highlighted is this repeat that goes AGG, AGG, AGG. This is a microsatellite. Microsatellites are usually found in non-coding regions or introns. The different variations of the microsatellite, or the different alleles, are when the DNA sequence has a different number of repeats. This usually comes about when during DNA replication, the DNA polymerase slips and accidentally adds or subtracts a repeat unit. And because it's non-coding, these stutters don't get edited out and they stay in the sequence. So here you can see we have different alleles. Each allele has a different number of repeats. And this makes the length of the DNA a different size. So here the first one has 8 repeat units, so the PCR product would be 41 base pairs long. The second allele has 6 repeat units, so the PCR product would be 35 base pairs long, etc. So with a microsatellite, there are usually many different alleles. This is really useful because this means that you only need a few different microsatellites, like 10 or 15, in order to have the resolution for individual identification, also known as DNA fingerprinting. Because you can distinguish individuals using microsatellites, these are quite useful for anything where you need to ID individuals. For example, in paternity analysis, population estimation, or even forensics. You can also use them for getting more information about different populations, for comparing things like genetic diversity between populations. Microsatellites are co-dominant, meaning that you can identify both of the parental alleles, and one parental allele does not mask the other. Remember back to codominance from our earlier talk about types of inheritance. Co means together, so you can see both parental alleles. Here, the brown plus the white equals brown and white spotted. That's how we remember codominance. The disadvantages to using microsatellites is that because they are in the non-coding regions, they don't tell you much about gene function. Also, if you don't have any genetic information about your species to begin with, microsatellites can be difficult to find and design primers for initially. The next marker is a single nucleotide polymorphism, or SNP. This is when we have a substitution of one base pair for another. As you can see here, we have 3 different individuals. The first individual has an A, the second one has a G, and the third one has a T. SNPs are quite common across the genome, and they can be found in non-coding regions like the microsatellites, but they can also be found in coding regions, so they might be able to tell you something about gene function, which can be useful if you want to know how the environment shapes genetic variation. So you can think back to the pocket mouse, how in this case there was a substitution which was a SNP between the 2 different forms, and the environment shaped that SNP and made it useful for the mice in the dark environment to have dark coat colors. Because a SNP is just a single nucleotide change, you can design really short PCR products. This is useful because that means you can search and find these markers in degraded samples, like old museum specimens where the DNA has been degrading for 50 years. Or you can also use SNPs to identify species, even by taking something like a water sample from a moving river and looking for the presence of small amounts of fecal DNA from fish. SNPs are also codominant, and so you can identify both of the parental alleles. One of the drawbacks of SNPs is that there is usually only 2 alleles, so an A turns into a G, for example. This means that you need many more SNPs, like around 50 to thousands, in order to identify an individual or to compare populations. This is doable, but can be expensive. Also, when you're looking for SNPs in the genome, you need to make sure there is variation. So for example, if you find a SNP where every individual in the population has an A and no one has a G, then that SNP doesn't actually have variation and isn't useful for that population. So the bottom line with SNPs is they can be quite useful for most applications, but you need a lot of them, and that can take serious time, effort, and money. The final marker I'm going to talk about is the amplified fragment length polymorphism, or AFLP. This is a more old school technique that's not especially common these days, but you do see it now and again. The way it works is you take a special enzyme called a restriction enzyme. This is an enzyme that is programmed to find a specific DNA sequence and always cut at that sequence. Here you can see the enzyme looks for where there is a sequence GAATTC in both directions, and then it cuts there. So to use AFLP, you take all the DNA from your organism and add 1 or 2 of these restriction enzymes. Here is a picture of this where you have 2 enzymes that were added, a red and a blue enzyme, and these arrows show you where they cut. The enzymes cut up the DNA into little pieces, and you end up with thousands of pieces of DNA. This many pieces can be difficult to work with, so then you add adapters that bind to the sticky ends of the DNA that enzyme creates. Here you can see the adapters as the red or blue segments on the end. To reduce the number of DNA fragments, you use PCR to amplify only those fragments that have both a red and a blue adapter. And this is called selective amplification. Finally, you run the PCR product through a gel or capillary, and you can see all the different fragments that split up by size. So here's a gel that shows AFLP results. Remember, each column is an individual, and you can see that many of the columns have the same bands, but the triangles are pointing out different alleles, where some samples have the band and some don't. This is where you have your variation. A more modern way to look at this is on the capillary, which shows you the same thing. So here each figure is a sample, and the peaks are the DNA fragments, and then those numbers show you the size and base pairs. If you look down this row here, you can see that each sample has the same first two peaks, but then for the third peak, the second sample is missing a peak. If there is a peak, that means that there was a cut at that site. If there is no peak, that means the enzyme didn't recognize the cut site and didn't cut it. The green arrows show you where there is variation in the alleles across the samples. Some samples have a peak there and some don't. And you score each of these peaks as a presence, so you would score that as 1. Presence of a peak is 1, or absence of a peak is 0. With AFLPs, the marker is dominant and recessive because the one allele with a cut masks the other. So what does this mean? Well, let's take a look at this in a smaller sample. So let's say that the letter C, the letter big C, stands for the allele that has the code for the cut. So if the sample is homozygous for the cut, it would have big C, and each allele will have that code for the cut, and it will be cut, and so it will have a peak. Now for the heterozygote, where we have big C, little c, one allele has a region for the cut, and the other doesn't. But you'd still see a peak here because some of the DNA would be cut. So in this case, the allele that has the code for the cut is dominant because it masks the other allele. And finally, this is the homozygote for the no cutting region. We have little c, and it's not gonna be cut, so you're not gonna have a peak. And the reason this dominant and recessive gets complicated in analysis is because it's hard to distinguish the homozygote from the heterozygote. Other than the dominance, another disadvantage is that there can be a high error rate, and it may be difficult to reproduce results when you try to do it in another lab. The good thing about AFLP is that it is cheap and easy to do, and you don't need to have any prior information about the DNA sequence, so it can be useful for an initial analysis. It's generally not detailed enough to use to identify individuals, so it can't be used where you need to distinguish an individual, for example, in forensics or paternity or population estimation. But it can be useful to compare different populations. So for example, someone in our lab looked at AFLP differences between the Louisiana water thrush. This is a bird that forages in streams and nests nearby the streams. So they looked at blood from birds that live near healthy streams and birds that live near streams with acid mine drainage, and they found that there were significant differences in the banding pattern between the two groups. So there are several different types of markers that you can use in a study, and each type has advantages and disadvantages. Ultimately, you need to know the question that you're going to ask in order to figure out what kind of marker you should use. So you think about things like, do you need to know individuals? Do you need to be able to identify populations? How much money do you have? Or these types of questions. And then from there, you can choose which type of markers you'll use in a study.